system as it is active in your body, how our body processes disease and that kind of stuff. So we're going to talk about the immune system in that aspect. The second half of it is talking about how we've kind of hijacked your body's immune system to do lab testing with it, and how we apply it to find diseases and to identify diseases using antibodies and using your body's natural immunity. And then the third part of it is kind of miscellaneous and assorted stuff that doesn't fit anywhere else. We're going to talk about the different kinds of diseases that are usually identified via serology. So in hematology, your white cells and your red cells, while they were separate, it all still kind of flowed together. This one's a little bit chalkier, okay? You do need to know a lot of things, and I'll be we'll be very careful to make sure that I stress as much as I can. There's a couple of terms and a couple of things you have to know. This first one is important. The next one that we're going to do, the antibodies and antigens, you need to know that one backwards, forwards, upside down, inside out, because that really is the meat of how we take care of diseases, and it's also the basis of how we use serology for testing. Okay.
Is that going to tie in with blood bank? Blood bank as well.
Oh God, yes. If you don't understand the next lecture on antigens and antibodies, blood bank will be a living hell. If you understand antigen antibody reactions, blood bank is just a game of where's the antibody, where's the antigen. That really, really will make blood bank for you a lot easier. Okay? If you, like I said, if you don't understand antigen antibody reactions, God bless you, because it's going to be really hard. And blood bank is one of those, some people just get it, and those people who get it are like, I don't understand what these people's problem is. And the ones who don't get it are just like, it just ain't clicking. So like you got the math gene or you don't got the math gene. I went ahead and I'm using Gene's old PowerPoints one more time. I haven't quite got a flow for what I want yet. Immunology terms, it's just a little bit of, I'm sorry, I'm really bad this morning. This is just really basic things that you need to understand in order for me to even talk about immunology. Immunology is simply the study of reactions of a host when foreign substances are introduced. Immunology is your immune system. It's what keeps you from, dying 5 seconds after you're born. Okay? You've got to have an immune system. Someone who is immunocompromised for whatever reason is going to have a lot more illness, is going to have a lot more problems. Immunity is simply the condition of being resistant to infection, whether it's because you've already had it, or whether it's because you have some sort of natural immunity, or whether it's because you've gotten a vaccine for it. That's just what immunity is. These two terms right near here, you need to circle them, star them, put big X's by them, highlight them, put flashing emojis on them, whatever you've got to do. These two terms right here are what are going to make or break you in a lot of our serology and in blood bank. And we're going to start with the antibody definition. An antibody is a protein that is produced by B-cell lymphocytes. We talked about that this summer. It is a protein produced by B-cell lymphocytes in response to a specific stimulus. An antigen is the stimulus. It is like the world's most circular definition. An antigen is what makes your bodies make an antibody, and an antibody is what is made when you are exposed to an antigen. The two terms don't come apart. Okay? They are interlinked perfectly. You will make a specific antibody to a specific antigen. And we will talk a lot more about antibodies and antigens in the next couple lectures. Natural or innate immunity is also known as passive immunity. These are things that your body has that are just kind of a general protection against all diseases. The largest organ, what's the largest organ in your body? It's part of the immune
Skin.
system. Your skin. If you don't have skin, all the buggies get into the good juicy stuff. Okay? But is your skin particularly against anything in particular? No. It's just sort of a general barrier. We also have antibodies, IgA specifically, which we find in our secretions. Normal flora is part of our immune system. People who are overly washing their hands and trying to sterilize their lives, they're killing themselves. There are millions and trillions and billions of buggies all over you there. They're supposed to be there. They have to be there. Okay? If you've got your nice normal staph epi and strep epidermidis, and they're colonizing your skin, there is no place for the undesirable things to move in. When you start making sure that you're totally sterilizing your hands, well then suddenly the things that are immune to what you're sterilizing with are going to come in and they're going to go, oh look, a nice open neighborhood, plenty of room for me. Okay. Acquired or cell-mediated immunity is also known as active immunity. And this is the body's response to disease or response to infection. It's what our body does. We mobilize our forces when we are invaded. So this is active. It's going to be against something in particular. This is passive. It's natural. It's innate. It doesn't really do anything. It's just there. Chemotaxis. Remember we talked about cytokines? Smiling on. Yeah, we talked about cytokines. I remember that exactly. Especially the interleukins. They're chemicals, right? They're chemical messengers. And basically what a cytokine does is it sends out a signal and it says, macrophages, neutrophils, why don't you come have some lunch? It's an invitation to a buffet. Right? We've got all these little bacterias here. Why don't you come in and help us out? It's a chemical system that lets the body know where the monocytic or the phagocytic cells need to go and where our lymphocytes need to show up in order to fight the disease. Diapedesis, basically just head is just foot. You've all seen, okay, we know how a red blood cell goes through a capillary, right? Well, white blood cells especially, because they are motile and active, okay, they can actually squeeze out between the lining cells of your capillary beds. They kind of ooze out between the actual structures on the outside, and we've got pictures later on, okay? But this is just where, yeah.
Is that all white cells or just a certain branch of white cells? That
Well, the only
actually-
ones that I know do it are the neutrophils and the monocytes.
The phagocytic.
The phagocytic cells, right. That's the only ones I know that do it. I know there are other cells that can diapedes, but the only ones I know that do it are those two. You remember when we talked about monocytes, what did I tell you about them? In your bloodstream. Are they active in your bloodstream? Not usually. They're just kind of hitching a ride to where they need to go. Where are they active? And what kind of cell are they in the tissues? Macrophages. Have you gotten that far with Jean yet? For pictures from macrophages? Okay. So they move out of the bloodstream by diapedesis and go to where the site of the infection is and turn into macrophages. Opsonization, it's something that attaches to a foreign substance to make it susceptible to phagocytosis. It's like salt. These don't taste very good. We'll put a little spice on them and ooh, yeah, we love those. Okay. It basically makes it tastier. It makes it more attractive to the phagocytic cells. As I referred to a little bit earlier, our external dispense system, these are things that keep the bacteria from getting in. There are structural barriers that prevent the infection agents. Unbroken skin, and that's very important that it's unbroken skin. Even microscopic breaks which is why, especially during the wintertime, if you're washing your hands a lot, you need to keep them nice and moisturized, because otherwise you'll wind up with microscopic breaks in your hand, and just because it's cracked from being dry. The pH of skin is about 5.6. It's just a little bit acidic. Not horribly, but just a little bit. The mucosal membrane surfaces line your nose. Okay, that snot's not just up there to make us miserable. The reason it's there is to trap things so that they don't get further into your body, which is why when you have an allergic reaction, you produce so much more of it. Our skin has lactic acid in the sweat that is produced. The lactic acid is produced by the working of our muscles, and some of it is excreted in our sweat. And what does that do? That makes it just a little bit acidic, which makes it unpleasant for bacteria. Okay. We have fatty acid in our sebaceous glands. Again, normal flora is also part of that. If the foreign substance is able to get through the external system, then the internal system gets activated. I also want to add to this one, sorry. What's in your stomach?
Hydrochloric acid.
Hydrochloric acid. Most bacteria can't live inside normal hydrochloric acid. Okay, so it not only digests our food, but it also kind of helps keep things from getting further in. Okay. Our internal defense, and again, you need to keep in mind, this is non-specific. This is passive immunity. Okay? It is not specific to necessarily to some one particular thing. We produce acute phase reactive proteins. Basically, the inflammatory response is what's supposed to happen when you get exposed to diseases. They're finding a lot of disease states themselves are a side effect of this inflammatory response. Okay. It is dangerous to your heart if you are in a constant state of inflammatory. If you are pregnant, a lot of times you'll have an inflammatory response just because, well, you're pregnant. Okay. If you're obese, you will see an increase in these inflammatory proteins. There's a zillion other things. But these can cause problems if they're there and they're not supposed to be. These are supposed to be something that kind of comes in, takes things over, cleans things up, and then go away. A persistence of these, it can show that either there's some disease state still ongoing or can in itself become a problem for you. These acute phase reactive proteins are produced by the liver in response to infection or to tissue injury. They serve as those chemical messengers to call in the troops to come in and fight the disease. They are released in response to signals from cytokines. Inflammation is an overall reaction of the body to injury or invasion by an infectious agent. Basically, there are 4 stages to inflammation. It's redness, or 4 pieces to inflammation: redness, swelling, heat, and pain. If you cut yourself, just a little cut, if you notice it kind of gets red around the cut, swells just a little bit. Even if you don't get a fever, the site will get hot, okay? And it hurts. Well, it's supposed to hurt so that you know you hurt yourself. Phagocytosis is the first step in the initiation, and this is just the engulfment and digestion of foreign particles by our phagocytic cells, which are neutrophils and our monocytes. Monophils, yeah. Our monophils, yes. So we're just going to call them that way you don't have to say 2 words. Both of these are phagocytic, and basically this is a natural behavior for any one-celled organism. How do cells eat? If you took basic biology, you know that they kind of sidle up and they go, hello, and then they sort of, here, come on in, and then they digest you, okay? So this is simply, our body has cells which have evolved to be extremely effective at phagocytosis. And there's the picture it's the basic, okay? We have our nucleus, and then we have these lysosomes. What do the lysosomes look like in our neutrophils? That's our granules. They're filled with a myeloperoxidase. Okay? It's kind of like a hydrogen peroxide type thing. And what does hydrogen peroxide do? It kills bacteria, doesn't it? Hydrogen peroxide is not good for most living cells. And basically the cell just goes over, forms a pseudopod around it, brings it inside. A phagosome is simply any vacuole which contains bacteria or foreign matter. Okay? If it is the neutrophils, these granules that are in there are going to inject cytotoxic chemicals and break it down. The monocytes are just simply going to digest it normally. Yes.
So, okay, that was, I think you just answered it. Monocytes don't have.
They don't have granules. No, they don't have the.
Okay.
Yeah. Diapedesis, we already talked about that. So step one, diapedesis is the first step of our phagocytic process. They got to get out of the bloodstream and get to where they need to be, don't they? Okay. So that's the first thing. They're going to leave the circulating pool, and they're going to wiggle their way over to where they need to be. Physical contact, basically, they're going to feel around. Oh, there it is. Okay. And then they're going to bring it in. Cytoplasmic granules and the phagocytic release enzymes, which digest the foreign particle. Then, when it gets all done, they spit out the leftovers. Okay, it's like they chew it up, get all the good stuff, and then spit the rest of it out. Okay? This is actually important because some of what they're spitting out is going to be used by our lymphocytes, and those little pieces of what were left over are going to cause an antigenic reaction and make us produce antibodies. So this is actually an important piece of this response. And then our immune response is going to recognize the bacterial debris as being foreign, and it's going to start producing antibodies.
Can they ingest as many bacteria as they can within their life, or?
Unfortunately, it's suicide duty.
That's what I figured. Like a bee.
Especially for the neutrophils. The monocytes, because they're just basically following their nature and just munching, they can usually gobble up quite a bit, but they gorge themselves, and all of the extra digestive activity does tend to destroy them over time. So yeah, unfortunately it is a suicide duty to go and take care of this. What are the acute phase reactant proteins? There's a whole huge list of them. Bonus points to anybody who can remember the one I told you about when we were talking about anemias. That was a problem for us. Ferritin. It's not on this list, but it actually is one of our acute phase reactant proteins. We're going to start with the others: alpha-1 antitrypsin, haptoglobin, fibrinogen. Fibrinogen is one of our acute phase reactant proteins. What do you think this is going to put somebody who's in chronic state of inflammatory reaction? What's it going to put them at risk for? If fibrinogen, I heard it, blood clot. I think I heard it from, yeah, blood clots. You've got additional fibrinogen. So it puts you at a higher risk of blood clots, which is why people with chronic inflammatory reactions often have heart problems, DVT, and other hemolytic problems. Okay. You also have ceruloplasmin and serum amyloid A. That's nice. The two that are really important to what we test for and what we do, testing for CRP. What test did I teach you in hematology that I said is the world's worst test that we cannot get rid of? Sed rates. We can't stop doing that thing no matter what we do. And some moron wrote a paper on sed rates and CRP, and it's, oh, look at this wonderful paper. Yeah, we've been telling you that from the lab for the last 25 years. Okay, CRP is an acute inflammatory protein that increases rapidly following injury It peaks at about 48 hours, and then once the inflammation subsides, it goes away. Sed rate basically is affected by an elevation of all of the proteins together, and some of these proteins hang around a long time. So it takes a long time for you to get enough of these proteins to increase your sed rate, and your sed rate will stay elevated for a long time after the inflammatory condition's long gone. So we have tried for years to get doctors to use this as a indicator instead of the sed rate. It increases sooner, and once the infection is gone or once the condition is over, it goes away sooner. So the sed rate tells you what happened last month. Well, maybe not last month, last week. What happened last week? This tells you what happened earlier today. And within 2 days, it goes away. If the sed rate's still hanging out and elevated, it could have been a long time ago. Right? That's why we say the sed rate is a worthless test. Right? We will see this elevated in bacterial infections. It is being used, there's a lot of new protocols out there looking for sepsis. That seems to be sort of the in thing right now, is we're trying to predict sepsis without just blood cultures. So we have a whole bunch of things they're using. This was one they've been investigating as an indicator. If you have sepsis, your CRP will be elevated, and exceedingly so. Yes.
Will the CRP be elevated for several days if the patient has bacterial infection, like a joint infection?
Yes.
That's been going on for 2 weeks, it'll stay elevated?
It'll stay elevated until the infection is cleared up, because the cytokines will still be calling the phagocytic cells in, and as the debris is set out, it will still keep stimulating that same response. So it's the CRP that was produced at the beginning is gone, but it's continually being produced because the infection is still active. Once the infection is no longer active, then it goes down, which is why it's a much better indicator, because it will stay up, and if it's still up, you still have infection. Once it goes down, you've got it under control. It may not be gone, but it's under control. Complement is a series of proteins, and we will talk a lot more about these later. You remember the cascade for coag? Well, we have one for complement too. It's shorter. But basically what happens is it's just like in the coag cascade. This one chops this, which chops this, which cleaves this, which binds to this, which does this. And the end thing happens is this stuff sticks on the cells that have the antigens. And it basically makes them yummy. It's an opsonicizer. It makes them more attractive to the phagocytic cells, so it makes them lyse. Okay. When we talked about cold autoimmune hemolytic anemias, remember those? This is what coats your cells. And because it coats your red cells, the body thinks that they're lunch and eats them instead of foreign bodies. Okay. But we will talk a lot more about complement later. This is diapedesis. It says escaping. It's not really escaping. It's getting there. It's actively going to find things. The increase, what happens in the inflammatory process, we have redness, okay? And that's because the blood cells engorge and bring more blood in. Okay? Warmth, the heat, is again because we're increasing circulation to the area to increase blood flow to the area, so that we can get these white cells to come out of our capillary bed and enter our interstitial space and take care of whatever's in our interstitial space. Okay? So they're actively crawling through. They squeeze out through the little holes between the cells. And then the cells pop. It's not like they break the capillary or anything. They ooze through and get out. Okay. Acute inflammation usually lasts up to about 48 hours. Okay? That's if you cut yourself, you get stung by a bug, something like that. It's going to come in, it's going to take care of it, it's gonna take about 2 days, it's gonna be all better, life's gonna go on. Chronic inflammation occurs if the infectious agent or other stimulus isn't cleared from the area. Chronic inflammation in some cases can be lifelong and permanent. Okay, this is a review, isn't it? Sure, we know all of this and we remember everything. Where are lymphocytes produced? Primarily in the bone marrow, but they're also formed in the extramedullary lymphoid tissue, such as the thymus. And they can clone themselves in the regular lymphoid tissue, your gut-associated lymphoid tissue, your lymph nodes. Okay? We have B cells and T cells. The B cells mature in the bone marrow. The T cells mature in the thymus. I know that's a toughie, but I think you can handle it, okay? Somebody was really, really thinking hard to name these things. Once your B cells have matured in the bone marrow, though, they exit the bone marrow and will set up colonies in the other lymphoid tissue. The T cells and B cells migrate to the secondary lymphoid organs. Periodically, they leave home and go on a road trip just to hang out and see if maybe they can't find something that shouldn't be where it is. They're kind of policing things to see if they can find things that don't belong. The spleen, which we talked about the spleen in hematology as regards removing old blood cells, but the spleen also filters out any damaged cells or any cells that have been eating bacteria. They'll have physiologic changes to them, and so the spleen will pull them out of your bloodstream if they happen to be floating through there. So your dead macrophages, your dead neutrophils, any organism which it recognizes as being damaged by infection. The lymph nodes basically are a collecting point for lymphatic tissue and lymphatic fluid. Lymphatic fluid. And all lymphatic fluid is basically your blood without the proteins and the cells. Okay? It's just a kind of proteinaceous, clear fluid. It circulates throughout your body. You actually have an almost second circulatory system. The circulatory system itself carries blood, but the lymphoid tissue carries this sort of tissue juice through, and that gets screened in the lymph nodes looking for antigens or foreign things that might have come in. We also have the malt, galt, balt is bronchial associated lymphoid tissue. That would be in the lungs. The galt would be in your GI tract. Your tonsils, your appendix, and cutaneous associated lymphoid tissues. I don't actually know what they're talking about for that one. But you have lymphoid tissues that are spread throughout your body, and their job is to be sentinels, to look for things that are there that should not be there. We talked about this also. Okay. This was, anybody remember how we check for this? The answer's in the next slide. We do flow cell cytometry. Does that ring a bell with anybody? CD markers, being able to differentiate cells based on their cluster designations, based on their surface proteins. 2, 3, 4, and 8 are our T cells. 19, 20, 21, and 40 are our B cells. And we talked about this is monoclonal antibodies. Anybody remember where we make monoclonal antibodies at? I warned you in heme, I bring stuff back up when I hit serology because it's appropriate. It is there. Anyone remember how we make antibodies, or how we come up with the testing antibodies for this. The old way was to inject a horse, a guinea pig, or a rabbit, or goat, and bleed them and purify the antibodies out of their blood. What's the new way?
It's not the banding.
Hmm?
It's not the banding.
Don't you just make them in a lab?
How?
A big
You're
vat.
right, in a big vat. Right, remember we take T lymph or B lymphocytes, we mix them with hybridoma cells, so we basically turn them into a cancer. We throw them into a vat of yummy stuff so that they will grow, and they basically voraciously go and they grow. You throw an antigen in there, and they go wild and produce large amounts of antibodies, which we then suck off the fluid that has this nice, clean, clonal antibodies. So monoclonal antibodies, they are always easy to predict. A polyclonal antibody, it may react with numerous antigens. When you're using the monoclonal antibodies, it will only react with one specific antigen. So you don't get as much cross-reactivity, you don't get as many false reactions when we're doing our testing. So on here, basically what happens is the little antibodies come in and they tag onto it. Fluorescent microscopy, we're going to talk more about this. This next two lectures are a little intertwined, cart and horse kind of thing. We talked, this is the active end, and this is our constant end, right? And we can attach something to the constant end so we can look at it. If we put a little fluorescent tag on there, and our cells that we're testing have the corresponding antigen, then we will see fluorescence. If our cells does not have the corresponding antigen, we aren't going to see that fluorescence. Okay? And we will actually be looking at these. This is an example of anti-nuclear antibodies. These are ANCA, anti-neutrophilic cytoplasmic antibodies. I don't actually know what that one is. That's a PANCA, and I don't actually know what that one is either. But we will be looking in a fluorescent microscope for that. Flow cytometry, we talked about this. It's an automated technique that uses monoclonal antibodies to identify the different lymphs. We send them through a analyzer, and it makes scattergrams. What's a scattergram? George. It's a two-dimensional representation of a three-dimensional space. This is where we have our three-dimensional box, and it puts things in, makes little clusters of them, hence the term cluster designation. And then we slice it to look at it because we don't do good looking at 3D things. We can actually print them out 3D, but it's really expensive. We can print them out in color, it's really expensive. So we get a 2D black and white. And this should be nice and familiar, right? Smile and nod. And this is simply a marker. This particular cell this one is negative for CD4, this one's positive for CD4, this one's positive for CD4. This is our CD4-CD8 ratio. We're going to use this to monitor our HIV patients, but we're also going to use this to define a T-cell lineage acute leukemia. Okay. Why are lymphocytes important? To identify lymphoproliferative malignancies. We talked about a lot of those, didn't we? To define immunodeficiency diseases. There's a variety of different immunodeficiencies that we can tell by CD markers. If my T cells are doing just fine, but I don't have enough B cells, I'm going to treat that type of immunodeficiency one way. If I have plenty of B cells and they're working just fine, but my T cells are missing, I'm going to fight that a different way. We can look and see if we've got some sort of unexplained infectious disease. We can type our B cells, especially our ATLs, those reactive ones. Okay? We can especially type those maybe to see if we have an increase in one particular line of cells, and that'll give us a clue as to what kind of infectious agent we're looking for. We're also going to, like I said, we're going to use that CD4, CD8 ratio to watch for acquired immunologic abnormalities such as HIV and AIDS. Okay. Our AIDS patients, we can pretty much predict how well they are doing based strictly on their helper suppressor ratio. We also use it to monitor transplants. We want to make sure we don't see a massive increase in T cells because that could indicate that we are either having a host versus graft, which is known as rejecting the transplant, or even worse, that we have a graft versus host, where active lymphocytes from the transplanted organ start attacking the rest of the body. It's not a good thing. Yes.
An organ that's been rejected, that can't be used for another person
No.
ever again.
No, it's been damaged. The body tries to eat it. So, facts about B lymphocytes, and they're about 20-25%, okay, 15-35, excuse me. Around a quarter are B cells. They transform into plasma cells when they secrete antibodies. Does anybody remember what's a plasma cell look like? That windswept one. And if it's got globules of immunoglobulins in there, what do we call it? Guillemot cell. It does all go together. Wow, Jean's good. She got dealing. I'm excited. I want to do this again because this is fun. Everybody woke up. Your T lymphocytes are somewhere approximately a little between half and 3/4 of your total lymphocytes. When they get exposed to an antigen, they become large activated T cells. Now, this doesn't mean they're necessarily ATLs, it just means they get big. They produce lymphokines, which stimulate B cells to produce antibodies. They also play a role in graft rejection. Our T cell subsets are CD4 and CD8, and what you really should have is about 1/3 CD8 and about 2/3 CD4. Okay, so you should have about a 2-to-1 ratio in a normal, healthy individual. Our T cells, basically, to get it very generally, our T cells are active in fighting off large, weird things, cancer cells, fungal elements, parasites. Our B cells are going to be much more active in things like bacterial infections, viral infections, by producing antibodies. So cancers and weird things are what we see in our HIV patients, because that's the stuff that, nobody ever gets histoplasmosis except HIV patients, okay? Just because that portion of their immune system is not functioning well. NK cells, these make up a small portion of our lymphocyte population. And how do we know they're NK cells? Because they're not T cells, they're not B cells, but they are lymphocytes. Okay? They initiate the cytolytic reactions against virally infected cells. They're found mainly in the spleen and in the peripheral blood. Can I look at a cell and tell you visually if it's a T cell, a B cell, or an NK cell? Not really. I might be able, if it's larger, I might guess it's a T cell. If it's those urophilic granules that confuse the bejesus out of diff students, right? Okay. That is usually an NK cell. But pretty much you can't, if it's a plasma cell, or if it's got immunoglobulin blobs in it, I can tell it's a B cell. Other than that, we will never get far enough to be able to master it. That was fun and exciting, wasn't it? that's just like I said, this is kind of an overview of the immune system. It's important that you know the difference between the passive and the active, know the various types of cells that are involved. A lot of our lining cells are phagocytic in and of themselves somewhat, so they'll be involved in stuff as well. I don't think we're gonna get all of this. I'm not gonna go over this one, but this is at the bottom there. This is the one that basically I put together for my MA students. So it's pretty much, yeah, it's pretty much these super basics,
So
okay?
the knows these or fail is not for us.
Oh no, the know these things or fail is for you. It isn't. What did I tell you you have to know? What are the two most important terms that we talked about?
Antibody antigen.
Antibody, antigen.
Okay.
I was kind of hoping to hint. I did put that one up last year, and there were a couple of students who studied from it for the first test and used part of it. Go through it; it certainly won't hurt. It does have a few more things on what the list are. What are you going to be responsible for? What's on the main one? But like I said, the serology for dummies is kind of helpful. Okay. Antigens and antibodies. Everybody needs to wake up and pay attention for this one. All right. Y'all want a break? Would you like to take 15 minutes? You can have it if you want. If you don't want it, we'll keep on.
Students are gonna break it, so we have 45, right?
Yes.
Now.
Huh?
15 minutes go by.
That or we'll just get done and I'll let you out early. Which way do you want to go with this? All right, antigens and antibodies. If we get through this whole one, yay. If we don't, that's okay too. What is an antigen? I'd like to start with what is not an antigen. A liver is not an antigen. E. coli is not an antigen. A red blood cell is not an antigen. A protein on the surface of a liver, an HLA protein, is an antigen. The OX19 inhibitor module on the E. coli is an antigen. The Kel molecule on the red cell is an antigen. Do you get the point? It's a macromolecule. It's going to be chemically rather large. It's going to be somewhere in the hundreds of thousands of daltons in size. But a whole organism is not an antigen. You don't form anti-eyeball antigens, but you might form anti-vitreous humor protein antigens. Okay, so you get where I'm going with that? All right, so it's a piece of, a part of a specific protein. One little thingy that's hanging off the edge of what you're exposed to. Okay. So it is a macromolecule capable of causing the formation of immunoglobulins. I'm going to kind of, you're going to take notes. I am kind of going to talk about things more than once in some of this stuff, hopefully just to make it bump your brain a little bit more. There are proteins, and we're going to talk about them in just a couple of slides, that are on each and every one of your cells. And they basically have a very specific pattern. They're called our HLA antigens, and we're going to talk about them. Okay? And it stands for human lymphocytic. I feel like a teacher when I get it right on the board. Antigens. Okay? And they have a pattern, just like your DNA or your fingerprints. Okay? What this pattern tells you is, this is me. I was like, we had Sam, got the little, remember Green Eggs and Ham? I am Sam. Sam, I am. Every single cell has a little sign that says, hey, it's me. And the important thing about an antigen is that it comes off of something that doesn't say it's me. You remember we talked about our lymphocytes flow through our lymphoid tissues, flow through the lymph system, flow through the bloodstream, looking for foreign bodies. They look for things that don't have a passport. They don't have a little sign that says, it's me. And if you have something in your body that doesn't say it's me, they'll attack it. They don't like it. So these compatibility antigens are how our body recognizes us. Okay? Lymphocytes are not smart. They don't have a brain. They are just single cells. They react to things based on chemical reactions and protein reactions. And what they react to are things that are not me. The antigen reacts with specific antibodies or sensitized cells that have been produced. The antibodies will come from B cells. The sensitized cells will be T cells. The antigen is called an immunogen if it elicits an immune response. We don't use that term a whole lot, except when we're writing fancy papers and we want to sound real smart. We're pretty much going to refer to them just as antigens almost all the time. The key part of the antigen that is recognized is the epitope. And each antigen, each epitope, has its own specific antibody. And they go together. And there's just, there's 1,000 different analogies that people use. It's like a lock and a key. Okay? The epitope is the lock, and the antibody is the key. They have to match up. If the tumblers don't match up, the key won't do anything, right? It won't hook up to it. It's like puzzle pieces. Each piece has to be, the two sides have to match exactly so that they hook together just right. So each epitope has its very own antibody, and each antibody has its very own epitope. Molecular weight, they'll be greater than 10,000 Daltons. For the most part, proteins are going to be our antigens. Those, because of their very structure, tend to be the most easy for our immune system to handle and to make an antibody against. If you remember when we talked about making antibodies, we talked about you've got the constant end, and we'll talk a lot more about that in a minute. And then you've got the wibbly wobbly end, the variable region. Well, proteins by their very nature are kind of globule. And it's very easy for that variable or mutable end of our antibody to form around that globule shape. So proteins, and they're very chemically large. So these are going to be pretty much the most common antigen. And it may be a portion of a protein. It may not be the whole protein. It may be just one piece of it. Then carbohydrates, our second sugars, also make fairly decent antigens. Lipoproteins will sometimes do it. But pretty much proteins, carbohydrates for the most part, we'll see some lipoproteins, and every once in a while the immune system just gets a wild hair and goes after something weird. It likes weird food too, I guess. Most important is foreignness. It's not me. All right. Plant protein is much more foreign than an animal protein, just because eukaryotic cells and eukaryotic cells are not. Eukaryotic and eukaryotic cells are not the same. They have a lot of structural differences, a lot of protein differences. They have the ability to be processed and presented by the MHC, the major histocompatibility complex. We will talk about that a lot. The pieces of the antigens must be complex to and presented to the lymphocytes. Okay? They don't do their own butchering. The MHC has to come in and kind of take it and hang on to it and say, okay, here you go. Now here, you make an antigen against this. I'll hold it and you take care of it. The key part of the antigen that is recognized by the immune system is our epitope, or our determinant site. And the number of epitopes per molecule is equal to the valence. And just like in chemistry, increased valence indicates increased love of each other. Okay, increased bonding, increased activity.
What did MHC stand for?
Major histocompatibility complex. We have a couple of different types of antigens. The first is a hapten. Pretty much what a hapten is, it's a wussy little hanger-on. It's not big enough to really do anything by itself. It has to be combined with some sort of carrier molecule so that it's big enough for the immune system to recognize. It's able to react with antibodies that are already formed. A lot of times it may be a portion of an antigen, of a full antigen. There's just enough of it that the antibody, it can't really grab onto it, but it can kind of hook it. It'll have just enough of a portion that matches up with that variable end that it can attach to the antibody once it's been formed against the full antigen. An autoantigen, we will talk about these when we talk about autoimmune disease, but basically, for some reason, your little sign that says, I'm me, the immune system thinks you're lying and attacks anyway. That or your sign got knocked down. And so the immune system says, oh, it's not me, I must attack it. Well, unfortunately, it is you, and it attacks it anyway. There is an entire slew of diseases that are called autoimmune diseases. We'll go into those when we talk about disease states. Alloantigen, allo simply means it's from another organism. Okay. Usually when we talk about alloantigens, we're talking about something that comes from another human. Right here in big bold letters, write blood bank. Because that's what we're talking about when we're talking about blood bank. When you receive a blood transfusion or when you are pregnant and exposed to blood from another individual, bless you, and exposed to blood from another individual, if there are antigens on their red cells that you don't have, your body can make an antibody against them. It doesn't always do so. Only about 15% of people are responders and actually make antibodies. But once they do, if you give it back to them, instead of going through and delivering oxygen, all of those cells are going to burst. And what do we know about free hemoglobin? It's not good for you. It's toxic. It will kill you. So this is how we form blood bank antibodies. Yes?
Is this what happened to the cows that caused mad cow disease when it mutated?
No. No. That's something different. Those are prions, not antigens. Heteroantigens just means it's from another species, a microorganism, a plant, an animal, and anything else. Okay? They're closely related in structure to the so that the antibody can cross-react. Antigen from another, they are closely related in structure so the antibody can cross-react. Anybody get that?
Related in structure, so they don't have cross-react. Antibody in one and the antigen from the other species.
I think what she's getting at is that, because she's talking about cross-reactivity, it might be that like if you are exposed to strep pyogenes, there will be some antigens that are in common with strep epidermidis. So these heteroantigens can cross-react with something that you've had before that's similar. Okay. Told you I'm redoing these. An adjuvant. An adjuvant is simply something that is added, a lot of times a salt, some sort of simple salt or metallic salt. That when it's usually something we add to it. This is something we see in a vaccine. Basically like aluminum salts, they help the hepatitis B vaccine to elicit a better immune response. They combine with our antigen that we're giving you, and they make it yummier. Okay? They make it more attractive to your lymphocyte. It's going to give you a higher, stronger, faster immune response, which is going to make you more immune in the long run. So it's going to increase your body's response, so we don't have to give you as much, okay, of that in the vaccine. Okay. I am going to skip MHC for today, and we will do it next time, because I have actually, this one I've been working on a lot, and I think I've got some better stuff for it. Okay. So we're just going to skip MHC for right now. We will come back to it. Okay, so just the next couple of slides. Hang on, and I will be bringing something else in. I have a hard time following her slides. Besides, I'm not finished watching the YouTube. Kidding, I am finished watching it. Okay, we're going to talk about what is an antibody. Okay, we already defined it as a protein that is produced by a B-cell lymphocyte in response to a foreign body. It is known as an immunoglobulin, and we refer to them as immunoglobulins a lot. In fact, as we usually refer to them as Igs. There's 5 kinds, and we'll talk about 5 different kinds. There's IgG, IgA, IgM, IgE, and IgD. The basic structures, they're gamma, mu, alpha, delta, epsilon. GAMED.
And are those different types of antibodies, or are they different parts
They're
of the
different. No, they're
antibody?
different classes. There are
Different classes?
five. There are 5 different protein molecules. We are going to look at each one. Okay. Somebody told me that they were told at some point that that's another acronym you can use. It's just you put 2 A's. You use an extra A. Okay. That antibodies cause damage. If it's easier for you to remember that, that's fine. But there's only one kind of A. But GAM, E, and D, so there are 5 kinds of antibodies. You will need to know the 5 kinds of antibodies. We're skipping major histocompatibility.
Okay.
And we're going straight to antibodies. Because I can't get through both today, and I'd rather get through antibodies and do MHC next time. So basically we have G, A, M, E, and D. Again, they're named for the Greek letters. We never use the Greek letters except on IgG. Anybody ever gotten a shot of gamma globulin? Probably not. That was a whole. Before we had a lot of the newer treatments that we have, if you got exposed to hepatitis or something, they would simply just give you a big old shot in the butt of gamma globulin, simply IgG, just as something that could go through and attach to the foreign stuff that was in there in hopes that it would stimulate your immune system and keep you from getting the disease. We don't use it near as much as we used to. Basically, we have a couple different regions. We have the FAB, which is our antigen binding site, and we have our complement binding site, which is the end region there. We have the light chains, the kappa and lambda chains. It's made up of one variable and one constant region. And again, I've got some other pictures here that We're gonna go over them and you guys can draw. I can't draw, so you guys. The two chains are hooked together with a variety of disulfide bonds. And right there in the center, where they go together, that's the hinge region, it's flexible. So these can stick together.
Real quick,
Yeah,
what are the chains?
they're the protein chains that make this up. So this is your kappa and lambda chains, and this is your gamma.
What exactly are the difference between them?
Just like the difference between any other two proteins, their structure,
Okay,
the sequence of amino acids that make them up.
okay.
Okay.
So this is our electrophoresis.
Yes, absolutely. Oh, that's right, you guys already had that. That's good. So when we look for kappa and lambda chains, we're looking for a spike in immunoglobulin, which is produced by excess plasma cells, which would be found in Waldenstrom's macroglobulinemia and in multiple myeloma.
Where's the disulfide?
The little black ones.
Oh, okay.
We do something, we use papain and pepsin. These are two proteolytic enzymes. And when we take those, what we can do is we can split this off into its various parts. And what they were able to do with that is figure out the structure and some of the function of what's going on with these. Hang on just a second. Isotype is a type of heavy chain. Its unique amino acid sequence is found in each type of immunoglobulin. So basically your IgG has one isotype, your IgA has an isotype. They each have a different protein that makes them a little bit different. The allotype means that it's the same basic immunoglobulin, but they have subclasses. IgG is the most common one that we look for the subclasses, and I think it has 5 of them.
This is has 4.
Oh, it has 4. I think there's 5.
Are there?
I think they kind of know, but 4 or 5. All I'll ever ask is, do you do immunoglobulin subclasses? An idiotype is a variable region of the antigen for each type of immunoglobulin. That's all we'll ever talk about these. All right. Almost want to go back to serology for dummies. We may in just a minute. IgG is a monomer. That means it's got one piece. Okay. It looks like a Y.
And it just has a heavy chain then, right?
No,
Oh, no.
no, they all have heavy and light.
Okay.
We're going back to serology for these. I have better pictures.
Oh.
We'll go back and talk about it all, but I've got, okay. The only reason I want to go back to this is I want to go with this. Okay. Because it starts talking on that slide about anamnestic response. So I want to talk about the response pattern of immunoglobulins. The function M is our first responder. We'll go over these in detail on each of those slides. This is our continued. Really, really strong. And repeat responder. This is found in secretions. This is allergies. And all of this is in the slides. I'm just going to, and this one, I can't find anything. I've been through every source I have. I've been through NIH. Nobody knows what the hell it does. They think it may help in the other one's response. Yes, Steve?
With G continued, what is continued increasing?
I will show you right here.
Okay.
Okay? So our primary response, this is time. This is when we get exposed. So what we get is we get a reaction with our IgM. This is important when doctors are ordering testing. If they're looking for a current or recent infection, we're going to look for that IgM. Half the doctors don't know that, but they just won't look. Shortly after the IgM, but before the IgM response goes away, we'll start making an IgG response. Notice it's a lot stronger. This is our continued and much stronger reaction. Okay? The other thing is that IgG comes down, but it never goes all the way away. We keep those memory cells that know how to make this particular antibody. So it hangs around down there. When we're testing your vaccination titers, looking for this. Yes?
In chronic condition, does IgM continually?
No, IgM goes away. The one that stays around is your IgG. The reason that our IgG stays around is our anamnestic response, which is on the other PowerPoint. I decided to do pictures. It makes more sense. Okay. If I am re-exposed, notice that my reaction is quicker and significantly stronger. Okay? What does this do for me? It keeps me from ever getting sick. So this is how a vaccine works. If I'm exposed to it, I make my IgM. I then make my IgG, which doesn't go away. And if I get exposed to it a second time, it's like rapid response. Okay? We know you're the enemy. And they squash them before they can actually get a foothold and become infectious and become a danger to us. So basically, if you're exposed again, it doesn't mean that your body the virus doesn't go in your body. It just means that your body is able to rapidly mobilize this secondary response, which is quick, fast, and effective. And it keeps you from becoming infected.
So, can you explain it for me again? You said that was, I understand it.
Okay.
When you said-
This is our zero. This is when we're first exposed. And it can be a vaccine, or it could just be that you have the disease. You make your IgM. Before you're done making your IgM, you start making IgG. Once your IgG reaches a certain level, your IgM tends to break off. And of course, there's exceptions to every rule here. Your IgM basically goes away. It's not as effective. Your IgG is much more effective, but it takes a little longer to get production going on your IgG. You get a much stronger response than you did with your IgM. Once the infection is gone, once you've got the foreign invaders taken care of, it's going to drop back down, but it's not going to go all the way away. You're going to retain a titer. You're going to retain memory cells that know how to make this particular antibody. So that if I get exposed a second time, remember this is time, so if you look at how long it took for my initial IgG response, look how long it took the second time I was exposed. Like nothing. And not only did it take very little time, but look how much stronger that second reaction is. Yes?
How would you define titer?
How would
What
I?
that means, yeah, the word titer.
How would I define titer? It is the highest dilution at which an analyte still reacts. So if I make a 1 to 2, a 1 to 4, 1 to 8, 16, 32, 64, and it reacts at 8, and it doesn't react at 1 to 16, then my titer is 8. It's the highest dilution at which you still see a reaction. Okay, yes,
So in the second
response.
exposure,
Mhm.
does IgM not come into play, or does it just have a smaller?
Oh, it probably does, but it's the IgG so fast that if it does come up, it comes up, goes away. You won't catch it unless you actually know.
Then
Will the second response have a titer like the first response?
No, this is your titer. This is your level. This would be your titer. This is your titer right here.
Okay.
So let's say this is maybe 1 to 1,000. This secondary titer is going to be more like 1 to 10,000.
So what's the definition of, like this, with memory cells? Does that have a name for it, or
This would just be a low titer.
no titer?
Yeah. The level, how much of the stuff you have to react. Okay? Titer is just a level. It's, instead of milligrams per microliter, we call it a titer.
So will it go down like that?
It'll go down like this. Okay? And this one will eventually, if you've gotten it a second time, you're going to stay at a much higher level for a lot longer. You will eventually, somewhere way over here, fall back down to a low titer. The one thing that can happen is, unfortunately, if our test only detects this is the lowest our test can detect, then we're going to tell you you're not immune because your titer is lower than what we can find. Okay. Titer is an old term for when we were doing things. Have you guys done the cold agglutinin?
Mm-hmm.
Okay, have you done the RF yet, the tube RF? Okay, the cold agglutinin, we used to do all of our serology reactions this way in a tube, and we made serial dilutions. So titer is a term doctors got used to. Modern instrumentation probably measures it in milligrams per microliter, but we still refer to it as titer just because this historically, that's what we referred to it as. So titer is just another way of saying how much is there, or the level.
So it will always increase more with each exposure, but it'll go back down to the same low?
I don't think it goes back down this far. You actually will,
Okay.
each exposure usually keeps you. But again, it depends on your particular immune system, the particular antigen. This is just a generalization overall. Each individual epitope can have a little bit different reaction curve. And when we get to hepatitis, there's gonna be bumps everywhere.
So a graph like this can be span over a lifetime.
This does span over a lifetime. You may never see this. Okay, I got my polio vaccine here. Hopefully, I will never be exposed to polio again. So you would never see this, right? Okay. However, if I was, the fact that my body has already been sensitized to it means that I will be less likely to get the disease. And again, it depends on the vaccine and a lot of other factors. But I'm not likely to get the disease. Or if I do get it, my son got the chickenpox vaccine. He also got the chickenpox. He had 9 chickenpox on his butt and his thigh. Because my mom, I went, Mom, she goes, it's chickenpox. I said, he's only got 9 spots. And he had the vaccine. Well, yeah, he got chickenpox. But he had such a mild case that basically he just walked around scratching for about a week. He loves when I tell that story. Yes.
So if you're always going to have the memory cells, why would you need to be revaccinated?
Because sometimes your titer drops so low and you have so few memory cells. If I give you a booster, remember what did we talk about? This is going to go up. Is this titer going to drop as far down? Probably not. So in some things, rather than give you a massive dose here, it's better to give you a little bit and then give you a little bit more and maybe even then give you a little bit more. Because every time I re-expose you, I'm going to increase the number of memory cells that are hanging around, and I'm going to increase your immunity. It's also why, especially with things like tetanus, if I get exposed again, they're usually going to give me a booster shot at that point, just to really kickstart my immune system so that it is going to really react quickly.
So that's the reason why a lot of people have 2 MMRs.
That's the reason why a lot of people have multi-step, or why the hepatitis B is a 3-step. Okay, and it's given at a specific interval because for that particular antigen, the response curve takes about 6 months, 3 to 6 months to fall. And so you don't want to give a re-exposure here, because that's more likely to give you a bad reaction, some sort of febrile, maybe even a seizure incident, because you've got too much antigen. So you really want this to come back most of the, if not all the way down. You know, you could give another one here, but you want to be careful not to overstimulate the first time. It actually is better if you let it fall, then make it rise, and then if you want to do it again, it'll go even higher. And again, it just will give you, it will, like a 3-step one, you'd go to this level the first time, the second time you'd be at this level, and the third time you might stay at this level. Does that make sense? So I said pictures make a lot more sense. Okay, pictures make a lot more sense. So I said serology for dummies. All right. There it is. Okay. So that's what this anamnestic response is. Okay? So all of that for one word. But I think the picture is definitely worth 1,000 words in this particular case. Okay, you're right. There are 4 subclasses. I thought there were 5. Oh well, I will look it up when I go home tonight. I'm wrong sometimes, you know. I'm wrong a lot. There are subclasses of IgG. We aren't going to learn the difference between them. They really are not that significant, except in a few very bizarre, very weird disease states. Just know that if the doctor asks for IgG subclasses, so sure, I've heard those before. But that's about all we're going to ask for. Something that is very, very, very critical and very important about IgG is that it crosses the placenta. This is a good thing, and this is a bad thing. It's a good thing because if mom is immune and she gets exposed, those antibodies are going to cross the placenta into the baby and provide immunity for the baby.
Does the baby keep that immunity?
Huh? No. Nope, because it doesn't have cells, it just has the protein. It just has, it's like giving you a big old shot of immunoglobulin, gamma globulin. It's not going to make you immune forever, but it might help you with that acute particular incident. It crosses the placenta, so it means that mom's antibodies can protect the baby, but it also means in the cases of hemolytic disease of the newborn, and you'll talk about this in blood bank again, if mom makes an antibody to the baby's blood cells, that can also cross the placenta. And how well do you think baby does if mom's IgG antibodies lyse the baby's cells? Not so good. And we talked about that. What did we make that has taken hemolytic disease of the newborn to almost 0? RhoGAM. And RhoGAM is an IgG antibody. It is a gamma, that's where the gamma comes from. It's gamma globulin. Okay. So it's important to remember that IgG crosses the placenta, IgM does not. IgG fixes complement, and we'll talk more about that when we talk about complement, when we talk about the MHC. It is an opsonin, but so are most of them. So basically this IgG will coat the outside of the cell that it is making an antigen against. Okay, so if I've got an antibody to E. coli, it's going to coat the outside of the E. coli anywhere that protein is at. And basically, it's salt and pepper. It says, here, come eat me. Look, I made it taste better. And it's going to release chemicals that are going to make our phagocytic cells want that particular organism. It participates in both agglutination and precipitation reactions. Remember I told you we divide this immunology into 3 pieces? This is the big piece that how do we test in lab for things? 99% of our tests are run using IgG. As a test method. And we will talk a lot more about that in the next section. IgM is the big boy. It's huge. It's honking. It's got 5 monomers. It's a pentamer. It can hook up to 10 different organisms at the same time. Okay? Goes in there and just, gotcha. Unfortunately, it's not near as stable. Okay? It only lasts about 10 days, and is only produced while the antigen is actually present. IgG will hang around when the antigen is not present, which is why I said when doctors are looking to see if it's an acute or a convalescent or a recovered, they're going to look and see if that IgM is there. It is a macroglobulin held together by a J chain, and we are going to go over each and every individual, learn how to draw the pictures of the antibodies here in a minute. It does not cross the placenta. It will fix complement. It is tasty. Okay. Now, like I said, don't worry about what it participates in the reactions. We'll talk about that when we hit the testing itself. IgA is a dimer, which means it's 2 monomers hooked together. It's found on the mucosal surfaces. This is our secretion antibody. Some people's are secretor, some people are secretors, in which case their secretions will have IgA. Some people are not, and so IgA will not be in their secretions. What does this mean? Some people have brown hair and some people have blonde. Okay. Some people have allergies, some people don't. Okay. It's just an extra passive immunity. Okay. It is found, it is an acquired secretory component from epithelial cells. Basically, it keeps the epithelial cell from being eaten by the invading organism. We're going to find it as a monomer in serum, so if it's in your blood, if it's going where it needs to go. But once it gets out into the secretions, it's going to pair up. Both hands and live happily ever after. Again, I don't know why they mention it's an opsonin, because all of them are. Spices. IgD, we find it on the surface of unstimulated lymphocytes. It might play a role in B-cell activation and differentiation, maybe. There's some research going on that. It's the second responder after IgM. You notice I didn't draw it in the drawing because nobody really knows what it does. Maybe it might help some of the other antibodies, and in there it might introduce them to the T helper cells that help the other antibodies make reactive cells. Pretty much, it's there. We can find it. It's protein. It's got a spike on the electrophoresis. Let's move on. IgE is also a monomer. It attaches to basophils and mast cells. And what's a mast cell? It's a basophil that made it into the tissues, right? Okay. It causes the release of histamine and heparin from the granules in the basophil. The biggest thing here is it is effective in parasitic infections. It attracts neutrophils and eosinophils. And I always remember that it's IgE in our allergic reactions because eosinophils and IgE make you sneeze. Lots of E's. Okay. The specific term, or the real term for allergy, is hyperimmune, which means overimmune. Your immune system, it gets in there and fights something that really doesn't need to fight. Okay. But IgE plays a big role in allergic reactions and also in parasitic infections. Again, we're in central Illinois. Which one do you think we're going to see more of? Yeah, the last lung fluke I saw. Yeah.
So an antihistamine, is that
The antihistamine.
using up the IgEs, or is that?
No, the basophils release histamines and the antihistamines keep them from action, keep them from acting.
And the IgE just kind of or I'm sorry, the IgE just kind of tells the neutrophils or the.
Yeah, it basically facilitates the reactions and facilitates the release of the chemicals from the granules. The eosinophils tell the basophils to blow up,
Gotcha.
okay? Again, I'm oversimplifying, there's, huge, massive, but for intents and purposes, that's good enough, okay? Monoclonal antibodies, we already talked about these, they're formed in the little vats, remember we talked about the little piranhas, you throw the thing there, they make these antibodies. And we use them for diagnostic testing. Aren't you excited? Food, all right. I will, like I said, I will do major histocompatibility next time. I've got some good pictures for it, and I don't have them with me. I have found some a little bit better. I also have, seriously, I have a YouTube video that I've been watching because it explains it a lot better than your book does. It's been a long time since I did histocompatibility, so I'll give you the link. Yes.
Are you doing homework?
Do you want me to?
No. But if you do, if you could put some weekly due dates.
If you want. I don't even know the reason.
Just making sure.
Yeah. That's kind of what Jean and I have kind of opposing views on that. She assigns it for homework because she knows people will do it, and I'm kind of of the opinion, I'm not gonna assign it. If you don't wanna do it, well, you're big boys and girls. You can do well on the test without it, fine.